Imagine Earth as a newborn planet still cooling down from its fiery birth. This was Earth about 4.5 billion years ago. Volcanoes erupted, spewing lava and gases into the atmosphere. The air was thick with methane, ammonia, and other gases we rarely find today. Lightning crackled across the sky. This was a hostile, alien world. Yet, from this chaotic environment, the miracle of life emerged. Scientists believe that the first steps towards life happened in this primordial soup of chemicals. These chemicals, energized by lightning and the sun's ultraviolet radiation, began to react with each other. Simple organic molecules like amino acids started to form. These molecules are the building blocks of proteins, essential components of all living things. The formation of these organic molecules was a crucial step. It showed that the basic ingredients of life could arise from non-living matter under early Earth's conditions. This process, called abiogenesis, is one of the biggest mysteries scientists are trying to unravel. Understanding how life arose from this early Earth is like piecing together a puzzle with many missing pieces, but every new discovery brings us closer to understanding the origins of life on our planet. In 1952, two scientists, Stanley Miller and Harold Urey, decided to recreate the conditions of early Earth in a laboratory. They filled a flask with water, methane, ammonia, and hydrogen to mimic the early atmosphere. Then they sent electric sparks through the mixture to simulate lightning. After a week, they analyzed the contents of the flask. The results were astonishing. Miller and Urey found that their experiment had produced amino acids, the building blocks of proteins. This groundbreaking experiment provided the first evidence that the building blocks of life could be synthesized from inorganic compounds under early Earth's conditions. It sparked a wave of research into the origins of life. The Miller-Urey experiment was a turning point. It showed that the basic building blocks of life could be created through natural processes. This experiment, while incredibly significant, was just the beginning. Scientists now had to figure out how these simple molecules assembled themselves into more complex structures, eventually leading to the first cells. The journey from simple organic molecules to a living cell is a long and complex one, but the Miller-Urey experiment gave scientists a starting point, a glimpse into the possible chemistry of early Earth that could have sparked the origin of life. Life as we know it is built upon cells. These tiny, self-contained units are responsible for all the processes that define a living being. But how did the first cells arise from a world of non-living matter? This is one of the biggest questions in the origin of life research. One crucial step was the formation of membranes. Cells need a barrier to separate their internal environment from the outside world. Without membranes, life as we know it wouldn't exist. Scientists believe that early membranes formed spontaneously from fatty acids present in the primordial soup. These fatty acids can self-assemble into spherical structures called vesicles. These vesicles, although simple, represent a crucial step towards the first cells. They provided a protected environment for other important molecules to interact and evolve. Within these simple membranes, the stage was set for the emergence of more complex biological systems. The formation of vesicles was a significant leap forward, but another essential component was missing, a molecule that could store and transmit genetic information. This is where RNA comes into play. Section 4. The first cells. RNA, or ribonucleic acid, is a molecule similar to DNA. While DNA is the primary genetic material in most life forms today, scientists believe that RNA played a crucial role in the early stages of life. RNA has the remarkable ability to both store genetic information and catalyze chemical reactions. This dual functionality makes RNA a strong candidate for the first genetic material. The RNA world hypothesis proposes that early life forms relied solely on RNA for both inheritance and catalysis. RNA could have acted as a primitive enzyme, facilitating the replication of itself and other molecules. Over time, DNA, a more stable molecule, could have taken over the role of storing genetic information. The transition from an RNA world to a DNA-based world is still not fully understood. However, the RNA world hypothesis provides a compelling explanation for how early life forms could have functioned and evolved without the complex machinery of DNA replication and protein synthesis that we see today. The emergence of the first cells, whether they were RNA-based or not, marked a pivotal moment in the history of life. These primitive cells were the ancestors of all life on Earth today, from the smallest bacteria to the largest whales.